Okay, so let's continue with DNA, where my computer froze last time. Oh, I wanted to show you that, so Gertie actually allowed me to borrow her chew toy. So this is pretty much exactly what DNA looks like. On the outside, you have the backbone, which is made up of alternating molecules of sugars and phosphate. And then the important stuff is in the middle, the rungs of the ladder. Okay, so this is where the base pairs are. Okay, so that's what it looks like. What do you think, Pearl? Go get it, go get it. Okay, so let's continue with where we left off. So when we talk about how many base pairs that DNA, that organisms have, so for example, and I know I went through this, but DNA in a typical bacterial cell, they have about 600,000 base pairs, okay, which is still a lot, right? Nothing to sneeze at. But you guys in every cell that has a nucleus have about 3 billion base pairs, which is a lot, okay? So that's once again why bacteria can go through binary fission in about 20 minutes, whereas you guys, it takes one of your cells with a nucleus about 24 hours to replicate. So when we talk about the building blocks, okay, which I referred to as a base, the base pair actually is what we call, or the base pair is made up of a building block called a nucleotide, okay? And a nucleotide is made up of the following components. So it has a sugar, okay, component, which is shown here in blue. In DNA, it's deoxyribose. In RNA, it's just ribose. And then each nucleotide also has a phosphate molecule, and the backbone is actually made up of alternating units of sugar, phosphate, sugar, phosphate. And then you have the base right here. And remember, the bases are either A, G, C, or T. And so if I ask you a quiz question, what is the building block of DNA? The truly correct answer is nucleotide. And these nucleotides are always floating around freely in the cytoplasm of cells. There's always a lot of them to help the different DNA processes that are going on. So to summarize, okay, DNA is made up of building blocks called nucleotides. The bases are in the center of the DNA and they pair specifically. So A always pairs with T, G always pairs with C. And the base pairs are held together with hydrogen bonds. And because those are very weak interactions, the DNA can basically zip and unzip very quickly. And you'll find out when I talk about these processes that is absolutely critical to the function of DNA. So the end result, if we were to make everything pretty, it would look something like this. Okay, here's our backbone made up of alternating units of sugar, phosphate, and then the bases are in the middle. So if this is an A, this is a T, if this is a C, this is a G, and so on. And then the whole thing is kind of twisted, and that's what's called the double helix. In reality, if we look at DNA under an electron microscope, of course, it's not that pretty. So this is a bacterial chromosome. So this is bacteria isolated from DNA. I'm sorry, DNA isolated from bacteria, speaking in opposites there. And you can see it has kind of this banded appearance, but it's not pretty like the previous photo that I showed you. But that's what DNA looks like under a microscope. So where do we find DNA? So in eukaryotes, the majority of the DNA is found in the nucleus. Okay, so when I talk about these processes, I'm going to be referring to nuclear DNA. That includes everything, like protozoans, fungi, parasitic worms. Of course, you are a eukaryote. Now, you've probably heard about these little guys, which are mitochondria. And mitochondria do have a small component of DNA. However, we're not going to talk about that in the scope of this class. Okay, so in eukaryotes, the DNA is found in the nucleus. But any cell that's alive carries its genetic information in the form of DNA. Okay, so E. coli has DNA. Streptococcus pyogenes, which is the nasty little bacteria that gives us strep throat, has DNA. Okay, so every living organism carries its genetic information in the form of DNA. And the only difference, every organism, their DNA is made up of the 4 bases, A, G, C, and T. The only difference between you and E. coli is how many bases you have and what order they happen to fall into. So if we look at not only the nucleus, okay, but also in bacteria, the way that we can get DNA to fit inside cells is in the form of chromosomes. And so chromosomes are just really tightly coiled DNA. And it's DNA that's wrapped around proteins as well. So this is what we call a karyotype, okay, which means looking at the actual chromosomes. And this used to be done under a microscope. Now it's done automated, using a machine to do this. But this is what an example of a karyotype of human chromosomes look like. Okay, so human chromosomes, humans have 23 pairs of chromosomes, and we organize them or name them basically largest to smallest. Okay, so chromosome pair 1 is the largest, and then they get smaller and smaller until you get to chromosome pair 22. And then in humans, chromosome pair 23 are called sex chromosomes, and this is what determines our gender. Okay, so if a human has 2 X chromosomes, they are a female. If they have an X chromosome and a Y chromosome, they are a male. Pairs 1 through 22 are referred to as autosomes. And autosomes just mean basically they're not sex chromosomes. They don't determine our gender. So all of your other genes and genetic information is on these chromosomes. Okay, anything from eye color to hair color to your liver enzymes, your cardiac enzymes, all of the genes that code for those types of traits and characteristics are located on chromosome pairs 1 through 22, which are autosomes. Okay, but one of the things I want to make clear is that bacteria also have a chromosome. Okay, however, it happens to be just a circular chromosome, and it's what I actually showed you right here. Okay, so this is still a chromosome. Chromosome just means tightly coiled DNA, but in bacteria, it is a circular chromosome, whereas in, fungi and protists and you guys, these are what we refer to as linear chromosomes, but it's still a chromosome, okay? Because, for example, if you were to stretch out all of the DNA in one of the nuclei of one of your cells, that DNA would be 6 feet long. Okay, so to be able to fit 6 feet of DNA inside not only one of your cells, but inside the nucleus of one of your cells, it has to be really, really tightly coiled. And that is why we have the appearance of chromosomes. It just means tightly coiled DNA. Okay, so in humans have 23 pairs of chromosomes for a total of 46. And you get, for each pair, you get 1 chromosome from your mom and 1 chromosome from your dad, okay? But it's independent. That is the process of meiosis, which I'm sure you learned about in your previous biology courses, okay? So half your DNA comes from your mom, half your DNA comes from your dad. Okay, so I'm going to stop there and get this uploaded, and then we'll pick up here.